Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Today I'll present about the diversity of marine phototroph. Energy flow are very important to the global environment change, preservation, protection of the living marine resources, and also to the diversity of marine microbes. It is also basic of the food chain in marine environment. Currently, study was found that energy flow through the microbial loops are too important. So, in traditional understanding, on 1970, there was found that the diatom and flagellate is the primary producer at that time. It is because that organism was trapped in the plankton net. And also, these organisms are numerous in the water quality, usually at eutrophic environment and the oligotrophic environment. Current study shows that by using deoxyribonucleic acid and detection and flow cytometry was found that there is a phytoplankton that is more smaller than the plankton net. In recognition of PICOCYANOBACTERIA dominance, around 1917 there was found by using fluorescence microscopy technique and electron microscopy technique. There was found that unicellular cyanobacteria, which is Cyanococcus species. This species usually found at tropical and subtropical region. In 1918, there was found by using the high performance chromatography and also by using flow cytometry. There is found parts of the PICOCYANOBACTERIA, which is Cyanococcus species. There is contain the vinyl A that can absorb 3 times more compared to the chlorophyll A. However, this species doesn't contain picobilipigment that will use as a typical light harvesting antenna. For the Prochlorococcus, it also can undergo OXYGENIC photosynthesis, and also it contains the vinyl A and B that can absorb the ocean blue light pretty well. In discovering of ubiquitous phototroph in early 2000 series, there was found 2 bacteria that undergo photoheterotroph, which is APB and exogenic photosynthetic bacteria and proteorhodopsin. Okay, APB, well known as aerobic and exogenic photosynthetic bacteria and anaerobic and exogenic photosynthetic bacteria. For APB, which is aerobic and exogenic photosynthetic bacteria, it was derived from sulfur purple bacteria, and it is also a photoheterotroph. Basically, we will know that we'll say aerobic. It is live in aerobic environment. However, for anaerobic and exogenic photosynthetic bacteria, it live in anaerobic environment. However, APB are quite similar with APB, but then the APB usually it will use oxygen to photosynthesis, but then it's not to ANPB. In addition, for APB, it can reach until 30% of primary production from that bacteria. However, it was negligible on global scale, but then it still contribute as a primary producer. Okay, another one bacteria that can undergo photoheterotroph, which is proteorhodopsin. Okay, rhodopsin actually as a light sensor in marine proteobacteria, and it is contain retiolopsin and also can contribute ATP. Okay, for example, bacteriorhodopsin is part of halobacteria in archaea, and basically it can transform light energy to transmembrane proton electrochemical gradient for synthesis ATP by using ATP synthase. This mechanism was restricted in special archaean bacteria distributed extremely in salty environment. But then, by using metagenomic analysis, they have detected that there was large amount of rhodopsin gene are in E. coli, and it can synthesize ATP and the like. Thus, it clearly that proteorhodopsin can be as a primary producer. Okay, next, for oxygenic photosynthesis using infrared light, and oxygenic photosynthesis are basically used in light harvesting, light transfer, and it is important in photosynthesis. Okay, in oxygenic photosynthesis, it will in quantification based on the chlorophyll A. Besides, range of chlorophyll A will determine as a photosynthetically active radiation, and it will evaluate the light strength. In 1996, there was found that there is a chlorophyll D in Acariochlorasis marina. Chlorophyll D is used as a shifted chlorophyll, and it can absorb far red light distributed in coastal area. It also can use as light harvesting antenna and as chlorophyll interaction center. Besides, chlorophyll D are equivalent with chlorophyll A. In 2010, there was found that there is a chlorophyll F from Cyanobacterium on stromatolite that is another redshift chlorophyll, and it can absorb around 720 nanometer and also far red light around 760 nanometer. Okay, next we'll turn to the strange phototrophic microorganism. Okay, the cyanobacterium like UCYNA was revealed by metagenomic analysis of phasing bacteria in marine water. Okay, UCNA, it is a particle that contribute nitrogen cycle in pelagic zone. Okay, normally cyanobacteria can grow by oxygenic photosynthesis and respiration and carbon fixation. However, UCYNA was lack of photosynthesis too, which is rubisco. However, UCYNA was lack of the photosynthesis too, rubisco, TCA, which is tricarboxylic acid, for respiration. Therefore, it might be symbiont in some organism. Besides that, OXYGENIC contain photosystem 1 that can synthesize ATP and chlorophyll that can contribute nitrogen cycle in pelagic water. Thus, it will contribute the transformation of the light energy for chemical energy. Thus, it will contribute the transformation from light energy to chemical energy. Okay, lastly, we'll turn to diversity of light energy transformation system and reconsideration of photosynthesis. From previous study, there was saying that phototroph is organism that use light as energy sources, and is classified as a photosynthetic organism that can undergo photosynthesis. For primary first student, they say that it is an organism that can synthesize organic compound by using inorganic carbon. In simplest way, they saying that primary producer and photosynthetic organism and phototroph is quite similar to each other. However, current study shows that phototroph is all organism that can synthesize ATP by using light as a source of energy. But then for primary producer, it is an organism that can convert inorganic compound to organic compound by using light energy, and also it can produce ATP to marine environment. that's all from me. Thank you.